One of the challenges with this is in some places such as sub-Saharan Africa, not everyone can have access to these foods because of the cost and transportation. And you can also think about having a pill, okay? Something as trivial as taking like a daily vitamin. Even that has a cost, and with creating it and transport, and not everyone will have access to it either. In the US, a lot of our food is fortified with, if you will, nutritional components as it's being milled, as it's being processed. Depending where food is being created in this place, such as sub-Saharan Africa, the infrastructure may not even be there. So what I'm going to be talking about a lot today is a concept called biofortification. And this is using plant breeding to improve the nutritional quality of crops. So I'm not saying that this is a panacea that's going to solve global nutritional challenges, but I will propose to you that these 4 pieces in combination could perhaps complete the puzzle one day. So why are we talking about biofortification, and what is it really going to do? So when we think about this, this is a technology that is really focused on the rural poor, and it has the potential to be sustainable and cost effective. Because how incredible is it to generate and create a seed as a plant breeder? Then you can give that seed to a smallholder farmer, and they can propagate that for the next 5 to 10 years. That is immensely powerful. So just the whole concept here, and as we walk through this, it almost becomes very intuitive in the sense that what we're trying to do is find the genes that are controlling nutritional quality. It can be a trait such as grains, if it's maize or wheat. It can be a trait dealing with the leaves if you're breeding for crops, such as like brassicas. And the idea here is to find superior alleles in these breeding populations that are controlling this nutritional composition variation, and to select on that in the breeding populations over time, so then you can have, and the key part here is have a locally adapted variety for whichever target region of the world that you're breeding for. So a large part of my research is focused on maize. But why maize? Maize is very much a global staple crop. In the US, it permeates our lives if you think about it. But in places such in sub-Saharan Africa, it's more than just being in the food. It is almost like a way of life when almost half of the calories are coming from this crop. And sadly, a lot of the maize varieties that are consumed in countries such as Kenya are not nutritionally dense. They're low in provitamin A, vitamin E, iron, and zinc. So this is something that we can perhaps address through plant breeding, and this is what my colleagues and I have been trying to work on for the past 10 plus years. Maize, in terms of its population size, where it evolved, is one of the most diverse species in the world. If you take any 2 maize inbred lines, it says diverse as comparing a human to a chimpanzee. Tremendous genetic diversity. 10 times more variation than any human being in this room. But the question is, how do you harness that with all of these modern tools of plant breeding that we just heard about? So one of the traits I'm going to focus on a lot in this talk is vitamin A deficiency. So let's look at the prevalence of vitamin A deficiency in the world. As you see, in India, Latin America, and sub-Saharan Africa, that's where a lot of the vitamin A deficiency populations are. So let's look at some of the quantitative values here. For the 7 million pregnant women and 127 million preschool-age children are vitamin A deficient. So that is a large segment of this world's population. And vitamin A deficiency causes 600,000 early childhood deaths and blindness in 500,000 children each year. And what is frustrating for me as a human being and as a scientist is this is preventable. So why provitamin A? It is found in maize, but it's also found in very low quantities in the maize grain. So if we look at maize here and provitamin A, so in maize grain, after the provitamin A is consumed, is converted to vitamin A in the human body. So for the most of my talk, I'm going to be focused on beta-carotene. That is one of the most important provitamin A compounds because when it is ingested by human beings, it is cleaved and it has forming 2 vitamin A. So it has 2 groups, whereas beta-cryptoxanthin and alpha-carotene, when it's cleaved, it only forms 1 vitamin A. Okay? So for the most part, when we're talking about improving provitamin A content, we're really talking about increasing the amount of beta-carotene in maize grain. But I would be remiss if I didn't also mention that lutein and zeaxanthin are also important non-provitamin A carotenoid compounds that are very critical for human eye health as well. So we should also be trying to maintain and increase those as well. So with any trait that you begin to work on as a plant breeder, you have to ask yourself, how much variation is there in the world's germplasm? So this is one of the first questions that we asked in the first kind of 10 years of the project. So when we look at the maize grain and we run our HPLC on it and we want to see how much carotenoids are in the maize grain, and when we look at the composition, the vast majority of carotenoids in maize grain are zeaxanthin and lutein. So if you recall from the earlier slides, those were non-provitamin A carotenoids. Very important for eye health, but not provitamin A. So when we look at provitamin A, such as beta-carotene, we're roughly at about 5%. So it's a very small fraction of the total carotenoid compounds that are found in maize grain. And then when you look at, say, a world's collection of maize inbred lines from a tropical and temperate breeding programs around the world, on average, you're talking only 1.7 micrograms per gram on average. And these are with the white grain lines removed. So a lot of the variation that you're seeing in color is coming from lutein and zeaxanthin. So for the Harvest Plus target, which is one of the groups that we work very closely with in countries such as Zambia, it's a target of that's tenfold increase from, on average, what you will see in the average maize inbred line that's being grown. So the question you should be asking yourself then, wow, carotenoids are colored. Why can't you just not select on color? So this is a little game that I like to play. So here we have these maize inbred lines. So these ears on the left here, they're very orange, as you see. So they have high total carotenoids, 37.36. This one is kind of like a pale orange color, and its total carotenoid value is 8.48. But when you think about running HPLC and ask the question, how much beta-carotene is there? And is this going to track with color? When you look at that, this particular inbred line here has only 4.19, and this paler orange one has 5.93 beta-carotene. So what that is actually telling you is you have to use genetic markers to select for this trait. You can select for increasing the total carotenoid value, like we can select to make this very orange, but the beta-carotene is not going to always track with the orange intensity of the grain color. So then you have to ask yourself, what as we as plant breeders can do, and then that's finding the genes and the superior, i.e. the favorable alleles in maize populations, and then harnessing that and breeding for that in populations. And that's precisely what we've carried out. So there's this giant sledgehammer called the Maize NAM population, which is 25 diverse lines crossed to a common parent B73 for 200 recombinant inbred lines for each of the 25 families for a total of 5,000 recombinant inbred lines. So this is a great way to dissect complex trait variation if that particular trait is variable in this Maize NAM population. And for most traits that we have accessed or have tried to focus on, there actually is variation for it. So we have genotyped this at high coverage, the parents in HapMap v1 and HapMap v2. And we have also run HPLC on 2 years or outgrowths of the Maize NAM population. So with all the checks and controls for the 2 reps, we're pushing close to 20,000 HPLC runs. So this was a massive undertaking and definitely not something trivial to say the least, okay? And at the same time as we were doing carotenoids, we were also doing tocochromonols, which is also referred to as vitamin E. And we've also run RNA-seq on the Maize NAM parents at 6 time points. So then we were doing profiling essentially on like some 40,000 Maize genes in the genome across 27 lines times 6 time points on the grain itself after pollination. So this is a massive phenotypic and genotypic data set. So what we want to do here first is map the QTL. And one great thing with this system is we can jointly map across all 25 families at the same time. And this is called joint linkage QTL mapping, where you're in a sense harnessing the power of 5,000 recombinant inbred lines to QTL map. And that was with 14,000 SNPs. But now that is with the recent recombination that was used to create the Maize NAM population. But for fine mapping, we need to use the ancient, i.e. the historical recombination that has happened in the evolutionary history of maize. And this is where the genome-wide association study comes into play now. And this is to actually drill down, in a sense, fine map those QTL down to the actual genes. So you're not having fun unless you're doing GWAS with 30 million SNPs. That is when you actually have a rush and you begin doing GWAS and you crash a couple servers at the same time, okay? So this is really beginning to access the full potential of the Maize NAM population and what it was intended for, to really identify what the causative genes are and to eventually try to figure out what the causative variants are. So the first traits that we mapped here are the carotenoid traits. So this is running HPLC on all 5,000 recombinant inbred lines. Excuse me, my mouth is getting a little dry. So what this graph is showing here is the number of QTL per trait on the x-axis and total traits on the y-axis. So we mapped 8 carotenoid traits, and also the cumulative trait is all of these 8 carotenoid traits. So that's what we call total carotenoids. And all of this is on maize grain. So in total we found 103 QTL. And as you see here, the range is from about 6 QTL to 18 QTL here. And these traits were highly heritable as well, ranging from 0.75 to 0.95, and that's in the broad sense heritability. So now this also gives us an opportunity to compare the number of QTL that were found for tocochromonols, i.e. vitamin E. And as you see here, fewer QTL were identified for carotenoid traits relative to the tocochromonol traits. So what we can kind of draw from this, or kind of infer, is that the genetic architecture for tocochromonols is more complex than for carotenoid traits in maize grain. And these tocochromanol traits were also highly heritable as well. And just for comparison, where the tocochromanol traits vary from 12 to up to 22 QTL, such complex traits as flowering time, plant height, and leaf architecture can range from 29 to almost 40 QTL. So these nutritional biochemical traits in maize grain are less complex than some of the more polygenic traits that have been previously analyzed in the maize NAM population. When we think about the effect size here, when we look at carotenoid traits, and what you're seeing on this graph here is the effect estimate relative to the NAM average, and these are the QTL effect estimates for beta-carotene, so only a total of 7 QTL, and in this graph right here, it's the effect estimates for the 13 QTL that were found for alpha-tocopherol, which is the tocochromanol that has the highest vitamin E activity. And when we look at this, you can see on the y-axis here, for the carotenoid trait, it ranges, as you see, from 0 to 40%, and for the tocochromanol trait, alpha-tocopherol is ranging from about 0 to 5%. So what this is basically now saying is that the carotenoid traits have a larger relative QTL effects than tocochromanol effects, okay? So there are fewer QTL for carotenoids, and their effect size for the QTL are larger comparatively to tocochromanol traits in maize grain. So that's a take-home. So what does this all mean in an evolutionary sense? So what we can try to postulate here is when you think about a grain endosperm carotenoid trait, which really only evolved in the past 8 to 10,000 years, there are fewer QTL for these carotenoid traits and larger effect sizes because it was a shorter length of time for variation to accumulate. So when you think about the coalescent history of maize as an organism, and you think about the age of a particular trait and the age of QTLs, a trait such as carotenoids is really only 6,000 to 8,000 years old. It's a very young trait. But when you think about a trait such as drought tolerance and tocochromanals, where tocochromanals are critical to be in the seed bank, it's an important antioxidant, and seed has to have it to remain viable and germinate from season to season. So this is also an ancient trait. These ancient traits, the effect sizes that are large tend to be fixed early on. And then what's left are QTL effect sizes that are much smaller. So this is essentially what is happening for maize carotenoid traits. It's a very young trait, and it hasn't been under strong directional selection for a very long period of time. So there are actually large effect QTL still. And when you think about the genetic regulation and control of this pathway, there hasn't been enough evolutionary time for new QTL to really evolve to give precise control of this pathway in maize grain yet. So this is a very young trait. But we're trying to figure out what the underlying genes are for the QTL now. Thankfully for colleagues in Arabidopsis and rice, they have figured out a lot of the key biosynthetic pathway genes for carotenoids and tocochromanals. So then we can ask the question, do our QTLs that we found intersect with these known candidate genes in the maize genome? So for carotenoids, for the known candidate genes that are involved in creating the carotenoids or actually degrading the carotenoids, there are 34%. But for the QTLs that do not overlap, there are 53%. So that's for carotenoids. So when you compare it to tocochromanals, only about 13% of our QTLs overlap with a candidate gene, in a sense an a priori candidate gene from the tocochromanol pathway. And 2/3 are not co-localizing with any gene from this biosynthetic pathway. So what this is really beginning to tell you is we really don't know a lot about the control of these particular pathways in maize grain. But one caveat I would like to give, though, is that we didn't, if you will, quantify the embryo and the endosperm size of all like 20-some thousand grain samples, okay? So some of the QTL that we could be mapping could be related to grain size. So this is a very key caveat there, okay? So now we want to use the machinery of genome-wide association to try to resolve these QTL now. So the first trait, using as an example here, is total carotenoids. So we found a total of 16 QTL, and on the x-axis here is maize chromosomes 1 through 10. On the y-axis here on the right is the minus log p-value from joint linkage QTL mapping, and it ranges from like 0 to a max value up here. And those QTL peaks are the green circles right here, or the green bars. On the left axis here of the y-axis is the GWAS marker value, and it'll range from 0 to 1. And what this is, it's in a sense, like a subsampling procedure of the maize NAM population 100 times. And if a particular SNP is retained in a model 100 out of 100 times, then the value is a 1. If it's retained half the time, then it has a value of 0.5. So when we then look at these GWAS SNPs, and these are these pink triangles right here, and look to see which genes they hit, from what we can see, we can get 6 candidate genes that are hit right in the gene themselves. So we have a very good idea of what these particular QTL actually are. However, there's still a lot of unknowns out there, and so what we're basically doing is generating hypotheses for these unknowns, and sometimes they're in the recombination suppressed region of the maize genome, and there could be hundreds of candidate genes there. So if we wish to really figure those out, we're going to have to do some serious fine mapping, and it isn't just the hard part of creating larger populations. You have to fight the constraint of recombination where it's suppressed as well. So there are some QTLs that we will not be able to map very finely unless we want to do enormous populations to find the rare recombinants. So back to beta-carotene, though, which is a trait that was only controlled by 7 QTL. When we did the GWAS and we looked to see which genes were hit, we were able to figure out 6 of the 7 QTLs. So for a trait such as beta-carotene, we actually know what the vast majority of the genes that are controlling this trait in the maize NAM population. And one thing to keep in mind, though, the maize NAM population is just a sampling of a global collection of maize lines. So maize has a lot of genetic diversity. There could actually be other genes that are contributing elsewhere in all alternative populations, and there could be rare variants that are not included in the maize NAM population. So when I'm showing that there's only 7 QTL for this trait, that's the thing to keep in mind, that there could be other rare QTL out there that just are not captured in the maize NAM population. So now we can design a pathway level breeding approach since we know what those 6 genes are that are in a sense controlling provitamin A. And 2 of the genes was this DXS2 and 3. So you can think of this as like the precursors up here. So this is kind of like driving a car when it comes to pathway breeding. This is the fuel that you're putting in the fuel tank, okay? So we also hit Y1, PSY1. You could think of this as the gas pedal when driving a car. This is putting your foot on the gas because this is the first step into the carotenoid pathway. So this is, in a sense, pushing the flux into the carotenoid pathway here. But as you see here, there's a key branch point going from either alpha-carotene to beta-carotene. So what else do you need if you're driving a car? You need a steering wheel. And to get our steering wheel, we have a weak allele of this enzyme right here. So if we have a weak allele here, then the flux of the pathway is being sent down the beta-carotene branch here. So if you have a steering wheel and a gas pedal, then you have to have a brake, right? So our brake is the CRIT-RB1 gene here, and it's a high hydroxylase. And if you have a weak allele of this hydroxylase here, it increases the amount of beta-carotene, okay? So we can really begin using a combinatorial pathway level breeding approach for this provitamin A target trait. And currently in breeding programs for the past 5 years now, they've been targeting these two genes here. So now breeders can begin targeting these three genes to improve total carotenoid value and provitamin A. So one thing that we also wanted to ask the question was, where are these particular SNPs hitting in the genes? So when we looked at this, we were seeing that a lot of the GWAS signals are proximal to candidate genes or within putative promoters or perhaps enhancer regions. So then we could then ask the question, does differential expression underlie the genetic basis of these particular QTL? So in a sense, we're just asking the question, is this an eQTL? So since we've done like all this fun RNA-seq at all of these 6 time points across the 27 founder lines, what we can begin doing then is comparing these FPKM values, i.e. the like counts of these particular transcripts, to the allelic effect estimates that we have from the QTL mapping. So when you look at the x-axis here, this is the effect estimates from the QTL, and the y-axis is the log to FPKM value. So for this CRIT-RB1 gene, so if you recall, that's our brake, you can see a consistent correlation between the allelic effect estimates and the gene expression 12 days after pollination and 20 days after pollination. So this is in a sense really an eQTL, and this is how this QTL is working. If you have more of the transcript, you're going to have less beta-carotene. And the alternative is our fancy steering wheel here, okay? So it's the same Y and X axis here, but this particular QTL only begins coming on strong 20 days after pollination. So whereas the previous one was consistent over grain development stages, this QTL is really beginning to pick up just 20 days after pollination, and it's not really showing up early. So we are now beginning to understand the temporal component to these QTL as well. And with all of that information, since we have an idea of what the QTL are, so now we can begin doing genomic prediction and genomic selection with these particular QTL. And this is just a paper that we just published showing that when you know what the gene is for a trait such as beta-carotene, which is very simple and very tractable, it's only controlled by maybe at most 7 QTL, you can really begin to predict carotenoid variation in maize grain. And in this particular case, when you know what the QTL are, you can just target this QTL, and then that QTL targeting GP is as equivalent to testing with genome-wide markers across the genome. This is probably one of the few traits in plant breeding where you can actually do this by just targeting the particular gene because it's a very tractable trait. But if you're doing GP in a trait such as like plant height or yield, you're still going to have to be doing genome-wide because it's going to be highly complex and highly polygenic with a G/E component as well. But this is at least showing us that we are hitting the right genes, and then we can actually begin using those in breeding programs, and we can predict the performance of offspring in the following generation after crosses are made. And we are doing this research very closely with Harvest Plus, and orange maize is actually very popular now. So in the country Zambia, there are 10,000 smallholder farmers that are now growing orange maize. Because one key point here is that education and outreach is critical when you're changing food. So in these countries, for the past 100 years, or perhaps even longer, people have been eating white grain maize, and it's a strong cultural preference. So when you begin to introduce any new food, and us plant breeders, as I was saying, we want to invent the future, but people have to come along with us for the ride, okay? So for this project, this is where education and outreach has been very critical. And now, as I was saying, there are 10,000 farmers in Zambia that are growing orange high provitamin A maize, and we even have, like, if you will, a shortage of seed to even provide because there are a lot more farmers that wish to be growing this as well. Climate change. So here we talk about provitamin A, but we need to begin developing resilient crops. Because all of us plant breeders know that you may have something very valuable, but if you're targeting a certain production or environment, it needs to be resilient at the same time. So we need to begin thinking about asking the questions of, yes, we need nutritionally enhanced crops, but at the same time, climate change is upon us, and the crops that we are releasing need to be tolerant to, say, heat and drought stress as well. So then you can think about in the framework of genomic selection, where you're trying to fastly develop maize inbred lines that are nutritionally enhanced for the grain and also tolerant at the same time. So when you break down the different components of a genomic selection breeding scheme, you obviously have to train the prediction models, accelerate recombination, because that is the evolutionary engine of all plant breeding populations. You need to maintain enough variation in the populations. And the area that I've been targeting is increasing the phenotyping capacity, high throughput phenotyping. What does that really mean when it comes to improving for heat and drought stress tolerance? This is sadly the state of the art. When we think about contrasting the evolutionary rates between genotyping and phenotyping platforms here, in the very late 90s, there's a few of us who maybe still remember using capillary DNA sequencers, pouring all those wonderful slab gels. Now in 2015, we're really beginning to think about single molecule DNA sequencers. Let's look at height, something very trivial. People have been doing quantification of height in plants for a long time. In the mid-70s, this person was checking out the height of this cactus here with a steel tape. 2015, we're basically still using now a bar-coded tape in the field. So we really haven't come that far if we think about it, comparing it to all of these wonderful high-throughput DNA sequencing technologies. But what can we do to change that as plant breeders? And this is the it's a high-throughput phenotyping platform that I helped create when I was in Arizona as an ARS scientist. And what this fancy little tractor allows one to do is quantify canopy temperature, canopy height, and canopy reflectance as this tractor is driving through the field, and you're able to phenotype 4 rows at one time. And of course, everything is on GPS RTK precision, and this is where the antenna is, and this is where the GPS RTK receiver and radio is right here. So when you're driving through the field and you're collecting all this phenotypic information, you have all of the phenotypes linked to GPS coordinates, so you can deconvolute all that after you collect everything for these particular phenotypes. And what this is also allowing you to do is do phenotyping over the entire growing season. If you think of a trait such as canopy temperature, which you usually have a very short window to do your phenotyping for it, you can now do hundreds to thousands of plots for this particular trait, depending on the speed of your tractor when it's driving through the field. So this was first carried out in a cotton recombinant inbred line population, and this was grown on drip irrigation in Arizona. And it's great to do high-throughput phenotyping in Arizona for drought tolerance and also heat stress because it's hardly ever raining and it's always hot. And even if it will rain, you can do some quick calculations and calculate how much ET was lost, and then you can tweak your drip irrigation as well. So what we're viewing here is a heat map of this recombinant inbred line population of 100 plots. And what we see here first is the drought treatment. And if it's green, that means it's a hot canopy. And so this is rep 1 dry, and this is rep 2 dry, and this is like the range of temperatures for the cotton cultivars. And this is wet rep 1, so if it's blue, that means it has a cool canopy temperature, and this is wet rep 2 right here. So this is collected with those IRTs mounted on the tractor. So this is, in a sense, a heat map of the field at 1 p.m. in the afternoon for this recombinant inbred line population. 2 treatments and 2 reps for each treatment. So one of the first questions then that we wanted to ask, are we seeing change over time with this tractor system? So when you look at 7 a.m. in the morning, 10 a.m. in the morning, and 1 p.m. in the afternoon here, and this black line here is the ambient air temperature here, and this red line is the population average for the dry, i.e. the drought treatment, and this blue line is the population average here for the wet, i.e. the control treatment here. And there is certainly a time by treatment effect, so as you would imagine, as it gets hotter, the canopy temperature is increasing because the plants under drought, they're not having a lot of water, so their transpiration rate is decreasing because they're trying to conserve water. So there, in turn, their canopy temperature is also increasing. Here, this is just showing the actual distribution of the population for canopy temperature. And this group right here is the dry plots, the drought plots. This is 1 p.m. In the afternoon on August 12th, and these are the wet plots right here. And these are the 2 parents, TM1 and NM24016. So the wet plots had a mean of 32.3 degrees Celsius, and the dry plots had a mean of nearly 38 degrees Celsius. But the main thing as a breeder is these traits are actually heritable. So if one wanted to breed for canopy temperature, you can actually breed for it. And you should also be asking the question, is this repeatable from week to week? So this is on day 217 at 1 p.m. here, and this is on day 224 at 1 p.m. here. And this is plotting just the ratio of dry to wet canopy temperature. And as you see, between these 2 weeks, it has R-square value of 0.47. So it is pretty repeatable from week to week. But keep in mind, very constant climatic conditions in a place such as Arizona, and everything is being controlled through drip irrigation. In a sense, these plants are trained very well at this point in the growing season. But then this allows us and provides us with the resolution to do temporal QTL mapping. So we can begin asking the question, what particular QTLs are turned on at certain time points within a particular day, and then on a particular day through the growing season, which we would not have been able to ask these questions if we were just walking through with a handheld IRT going from plot to plot. So here at 1 p.m., day 217, 1 p.m., day 224, we have some of the strongest effect QTL here on chromosome 20, and then on chromosome 10 here. And what you're kind of seeing then is a pattern, and as it can be coming to light, is at 7 a.m. and at 10 a.m., the effect sizes are weaker compared to 1 p.m. in the afternoon. But we now want to ask the question for yield. Is this particular trait, such as canopy temperature, important for yield? So when you regress temperature onto yield, kilograms per hectare, we see an increased correlation here for the dry plots. So what this is basically saying is the increased leaf transpiration rate, i.e. cooler canopy temperature, even under drought, contributes to higher lint yield. That may be somewhat a little counterintuitive. But if you think about it, if the canopy temperature is increasing, what's happening? No transpiration, no carbon being fixed. No carbon being fixed, the yield is not going to increase. So these particular highly irrigated environments in places such as Arizona really selects for the risk takers. This isn't like the dry land agriculture for cotton on like the high plains. This is highly irrigated, okay? So the plants that take a risk and keep on trying to fix carbon, and they don't care, they're going to yield higher because they know I'm going to come there and turn on the drip tape, okay? So then you should also be asking yourself, well, why don't we see as like strong a trend under the wet? Because what I will postulate here is that the higher leaf transpiration rate is providing more of an adaptive advantage in terms of yield when these plants are grown under drought. And so we've been pioneering with the tractor. Now we're playing with small unmanned aircraft systems as well now. And there's some colleagues in my lab, and this is what we're now pushing forward at Cornell. After 6 months, we finally have approval from the FAA to fly over our maize fields at Cornell this summer. So just as long as we're under 400 feet, of course. And we've been also trying to create small handheld phenotyping devices for breeders in the field as well. So this is a device that I kind of call a tricorder, if you will, that can do canopy temperature, canopy height, and some canopy reflectance as well. And it costs less than $200. This is just a prototype, and we've been playing around with plants in the greenhouse. But I also think we've been thinking about high-throughput phenotyping from the point of view of doing very large fields. We should also be thinking about putting these low-cost, highly powerful handheld devices in the hands of plant breeders at the same time. Okay, and so this last slide kind of ties it all together. I now have a new project focused in Zimbabwe where we are growing these high carotenoid maize hybrids under heat and drought stress. and flying over with drones, so we can then begin to look at the consequences of how heat and drought stress perturb the carotenoid profile of maize grain. Because, you know, that's one of the important questions I think that is still unanswered is, how do these environmental stresses impact the nutritional quality of these plants that we have been breeding for? A lot of our phenotyping has been focused on plants that have been grown in Purdue, Cornell, places like that where the plants are very well maintained, okay? What happens if you begin to look at the type of environments that these plants will actually grow in, like a smallholder farmer's field? So this is where I've been carrying my work now. And there's a lot of other projects I didn't really have a chance to touch on, but in my lab we're also doing research on jet fuel, a really interesting plant called Waiuli. What do you use for Waiuli? You make tires with rubber. That's what Waiuli is used for. So got a jet fuel project, a tire project, and also doing research on oats. And this is funded by Pepsi Company because they own Quaker Oats. So here we're trying to improve the nutritional quality of oats as well. And I've also have a new project where I'm improving the nutritional quality of cassava, and also trying to look at to see how gender preferences have an impact on plant breeding in sub-Saharan Africa as well, because that is an unexplored question. We all know gender preferences are important, but no one really in plant breeding is addressing this. So I now have a new graduate student. We're beginning to see what are some of the traits that are important to gender now, and how do we incorporate that in a plant breeding program. Corn, of course, and dry beans as well. With that, I would like to thank all the funding agency, my great colleagues. So this is for the nutritional work in maize. Dr. Dean Della Penna and his lab ran a lot of HPLCs runs, over 20,000. Folks at Purdue, Torbert, growing out the maize population, and Ed did a lot of genotyping for HapMap V1 and V2. And these are my fantastic colleagues who have helped over time in the funding agencies with the high-throughput phenotyping work in cotton. And with that, I thank you very much for your time and allowing me to share the work that me and my group are doing at Cornell these days. Thank you.
All right, thank you very much, Dr. Gore, for that presentation. I think we have about 10 minutes right now for questions from the audience. We'll do a question and answer session. So if you have a question, we have a couple of volunteers with wireless microphones in the back. You can raise your hands, and we'll make sure that the people on the webinar can hear you. Also, for those of you listening in on the webinar, your questions are a priority to us as well. So go ahead and type your questions now into the chat box on the GoToWebinar control panel, and we'll make sure we get to you as well. All right, looks like Seth has a question back there.
Hello, Dr. Murray.
Dr. Gore, thank you. Quick question. So the sort of reductionist approach of using HPLC on the carotenoids and then using markers, you can measure differences, but have there been any follow-ups for in vivo studies to see if they actually make a difference to the human nutrition in the end?
Yeah, so that's actually being carried out by Harvest Plus, and they just published a paper that came out fall, and it's shown that it actually will carry over into humans as well. I don't have the paper off the top of my head, but I can find it for you and have it shared with you. But yes. Yep.
So I had a question on the provitamin A production stuff. You're limiting, or you're looking for QTLs that limit beta-carotene hydroxylase. Do you see in the seeds to reduce, that was the break part of it?
Yep.
Do you see any correlation with what's in the whole plant, not just the kernels? And do you run into problems with abscisic acid production with that?
That is a fantastic question because that is two questions that have been, in my point of view, unanswered. So for these particular genes, from what we've seen as we tweak them, is really only doing changes in the seed themselves, and it isn't having any changes from what we can personally see for the whole plant, right? One key point though is in terms of the yield or how well the plant will germinate afterwards. That's kind of why I want to begin testing it in sub-Saharan Africa under heat and drought stress, because I just feel like it really hasn't come in contact with that yet. And I just feel like for our role, testing these high carotenoid lines in that type of place will actually help address and answer some of those questions that I feel we haven't yet. Yeah. That we haven't seen yet. Yeah. Yep. But one thing though is pretty much as soon as you harvest the ear, there's already turnover happening in the seed, and there's a lot of breakdown as well. Yeah. Yep.
My question refers to increasing nutritional quality. How does that affect the germination, the yield of the subsequent materials? Because that's obviously going to be very essential as well.
Yeah, that's what I was kind of addressing here. In terms of yield, we have not seen any trade-off, and this has been testing through our Harvest Plus partners. But I would caution, I would like to see it being tested under heat and drought to see if there's any trade-offs, where when you're beginning to change some of those pathways that are tied in with hormones that are important for that type of particular growing patterns, to see what happens. But as of this point, we haven't seen any trade-offs yet. Yeah.
You mentioned that the target to increase the vitamin content is 17 microgram per gram, but the base is 1.7. So how far have you gone? Where is your new materials there?
Yeah, so Harvest Plus has released lines that are or I should say they have lines in the pipeline that are 20 and higher. So they have surpassed that actually now.
These lines are from the new technology or from the germplasm diversity?
Germplasm variation.
Thank you.
Yeah, yep. Is there any questions online or no?
I don't believe you.
Okay, just thought I'd ask.
We have time for a couple more. All right, Dr. Yu?
Mike,
Yeah.
have you compared using the prediction for those selected QTL within those specific regions to just the genome-wide randomly select sets of markers to see the prediction accuracy difference?
Yeah, we have that. That was included in our paper that just came out. So for that we had 8 QTL, right? And then we did 100 permutations, if you will, and just chose 8 other genomic regions across the maize genome, and it was a lot lower. Yes.
Go ahead. Mine's on a different topic.
Just a question regarding the levels of beta-carotenoids in maize. How does that compare to what the golden rice has achieved? And what are your thoughts on just the transgenic means to do the same thing?
Yeah, so golden rice hasn't helped too many people yet, as far as I know. So there has been some research in maize where they've used the transgenic approach, and they've been able to increase it very, very, very high. I would say almost cautiously too high, if you will. So this is, I think, one of the few cases where we don't have to take a transgenic approach, and we can just use the alleles that are already in the population. But at the point in time when transgenic approaches will be more widely used in food, we have the right genes and the right alleles for that to actually happen, I think. So, yeah.
I think we got time for one more question.
On a somewhat different subject, Howard Buffett commented in 2012 that his corn was out-yielded by soybeans, and he farms in a very good area near Decatur. What is your strategies on drought stress? What are you following?
So that's a really good question, because when you think about drought stress in the U.S. and compare that to like drought stress in sub-Saharan Africa, it probably isn't the same thing, okay? So for what I would think for a place where that's totally reliant on rainfall, you would want a plant that can basically use water more effectively over its whole growing stage. There's certain phases of when the corn plant is growing, say, then they need to use a lot of water. And there's certain phases when the plant won't need to use a lot of water. So then if you can breed for a certain genotype that can conserve some of that water in like the profile of the soil, and then only begin to pull it out when it really has to have it, such as like when it's flowering and at grain fill, then I think that's the actual type of genotype that I would breed for. Yeah.
All right, thank you everybody for the questions. Let's thank Dr. Gore one more time, please.